Hi everyone, I hope you're having a great day. So in today's lesson we are going to discuss two things. First one is going to be recombination, and the second thing is called non-dysfunction. Alright, so remember yesterday I asked you to answer a poll about what was going on in the meiosis diagram at metaphase 1. So what was occurring was this process known as recombination. To be specific, this type of recombination is called crossing over. Genetic recombination gives rise to new combinations of genetic material, which therefore gives rise to offspring that are genetically unique from one another and their parents. In each diploid germ cell, there are two copies of each chromosome, i.e. the homologous chromosomes. One copy of the homologous pair was contributed by the female gamete, i.e. the haploid egg, and so it's of maternal origin. The other homologous chromosome was contributed by the male gamete, which is the sperm, so it's of paternal origin. During meiosis, genetic variation, i.e. recombination, is ensured in two ways. The first way is through the creation of gametes that carry different combinations of maternal and paternal chromosomes. This is what we call independent assortment. The second way is by the exchange of genetic material between maternal and paternal chromosomes. This is what we saw in that meiosis diagram, and this is what we call crossing over. Alright, so we're going to talk about these two processes in more detail here. Alright, so for independent assortment, we know that in metaphase 1, the chromosomes are arranged in homologous pairs along the equator. In each pair, the chromosome of maternal origin, i.e. we're going to say the pink one, is oriented toward one pole, and the other chromosome of paternal origin is oriented towards the opposite pole. The orientation of a pair of homologous chromosomes is independent of the orientation of the other pairs. So, this means that if we had an organism with, say, diploid 2n equals 6, one possibility would be one maternal and two paternal chromosomes facing one pole, and two maternal chromosomes and one paternal chromosome is facing the opposite pole. Another combination would be all the maternal chromosomes facing one pole and all the paternal chromosomes facing the opposite pole. There are many combinations of possibilities for an organism that is 2n equals 6. In this diagram here, we have all the possible gametes that can be produced due to independent assortment for a particular organism that is 2n equals 6. So for extra practice, see if you can draw out these 8 possible combinations at the meta 1 stage, if you have time. Use, as a kind of a hint, use different chromosome lengths in order to better visualize the different possibilities, as they have done in this diagram here. You can see there's one really long one, one kind of medium one, and one short one. So you can definitely see the different possibilities of gametes that can be produced. And also notice here I made a little mistake. These ones that are kind of like darkish purple, that's supposed to be pink. I tried to draw pink on the outline. So realize that those are pink. Okay, those 3 right there. Alright, so that's independent assortment. Now we have crossing over. So during prophase 1, we have seen that chromosomes synapse together while the chromosomes are lined up side by side. Non-sister chromatids may exchange pieces of genetic information. This switching of genetic material is what we call crossing over, which is what we definitely saw in that diagram yesterday here of the switching of colors. A section of chromosome that is crossed over may contain hundreds or even thousands of genes. Therefore, the result of crossing over is individual chromosomes now contain some genes of maternal origin and some genes of paternal origin. Although in this diagram we only see one section being crossed over, But it's important to note that crossing over can occur at several different points along the non-sister chromatids. Okay? All right. So both of these recombination events, independent assortment and crossing over, leads to the great diversity of individuals and species, and which is what makes you different from your siblings, your parents, your friends, anybody in this world. All right, so that's recombination for you. If you need to, take a little break. I know that was a little bit long, so take a little break and return and we'll talk about non-dysfunction. All right, so hi everyone. So now we are going to discuss non-disjunction. So this occurs sometimes when chromosomes or chromatids do not separate as they should during meiosis. Non-disjunction occurs at either anaphase 1 or anaphase 2. All right, so let's discuss anaphase 1 first. So in anaphase 1, non-disjunction occurs when the homologous chromosomes do not separate to opposite poles, and one entire pair is pulled towards the same pole. This leads to two abnormal gametes that have one extra chromosome and another pair of two abnormal gametes that have one less chromosome than they need. So we have all four gametes are wrong in non-disjunction at anaphase 1. So at anaphase 2 then, this is when non-disjunction occurs when sister chromatids do not separate to opposite poles, and both sister chromatids are pulled towards one side. So as you can see here, normal meiosis 2 has occurred, producing two gametes that each have one, which is what should be in the gamete. But here, non-disjunction occurred, and two of the chromatids were pulled to one side. So we have one gamete that has an extra chromosome and one gamete that has one less chromosome than it should. So when one chromosome is lost and a gamete is missing one chromosome due to non-disjunction, it is called mesonomy, right? So that's here, or these two gametes here, or this gamete here. So individuals who have lost a chromosome, they can have a syndrome called Turner syndrome. And in Turner syndrome specifically, they are missing an X chromosome. So people who have this disorder have female sexual characteristics which are under development, which makes kind of sense because they only have one X chromosome instead of two. So when an extra chromosome is gained, so in these two gametes here we have an extra chromosome, and this gamete also we have an extra chromosome, this leads to trisomy. So trisomy occurs in individuals who have Down syndrome. And they were born with an extra chromosome at the autosome chromosome 21. So you can read up on the effects of Down syndrome in your textbook. All right, so that is it for this week's lessons. Remember to check out all the information on the website for today's lesson, and don't be afraid to send me a shout, email, or text if you have any questions, and I will see you all tomorrow. Happy Thursday. Bye-bye.